It doesn't even have to directly involve the mom and dad. There's a very famous case of Sally Hemings and Thomas Jefferson. Does anybody know that story? So Sally Hemings was a child that was born. Sally Hemings was a slave of Thomas Jefferson. And while Thomas Jefferson was president, she gave birth to, I think, 3 or 4 children that the rumor was that Thomas Jefferson was the father of. And went for many, many years. People didn't know were they not. And in the past 15 years, they've actually done DNA genetic testing on the descendants of Sally Hemings and Thomas Jefferson to ask the question, can we determine if Jefferson was in fact the father of these? And it was determined that A. Jefferson was. Whether it was Thomas Jefferson or his brother is open to question. But the mystery over hundreds of years of which was the father of Sally Hemings' offspring has been answered, which is a pretty remarkable thing, I think. So, okay. All right. Let's see. Genomics, proteomics. I only want to say one thing here. I want to say something about microarrays. Microarrays. Microarrays are kind of a high-tech way of doing 2D gel electrophoresis. Okay? When we did 2D gel electrophoresis, what did we do? We isolated proteins, we separated them on the basis of charge, and then we separated them on the basis of size. So we got a spot for every protein, right? Another way of asking the question in terms of what genes are being made is not to look at the proteins, but to look at the RNAs, because the RNAs code for the proteins. Now, this technology is kind of mind-boggling. To do this, there are little chips that are made. The chip is a little piece of glass that has some DNA or RNA molecules stuck onto it. The molecules are stuck in the form of a grid, and a given grid might have 100,000 different individual sequences planted on that grid. Each spot on the grid, let's say top left, has thousands of identical copies of one sequence. The adjacent one has thousands of copies of another sequence. The adjacent one has thousands of copies of another sequence. And you know what the sequence is in each case. You pick the sequences because they correspond to genes of that organism. So you might have a gene for the DNA polymerase up in the top left corner. You might have a gene for a kinase up in the upper right corner, and all kinds of other genes scattered throughout the middle. You know the position of every gene on that grid. Okay? Now, you take and you have a person who had a liver cancer, and you take that liver and you grind it up, and you isolate the RNA, the messenger RNA, out of that liver. You treat it with a chemical so that it's got a color. Okay, so you put a red dye, for example, on the messenger RNAs from this person's liver. You then add the messenger RNAs to this spotted plate and let the complementary sequences come together. You wash it off, and then you ask the question, where are the red spots? Okay? You look under a microscope, and there's actually, this is very automated. You can look under there, and the microscope will look under there, and you'll take a picture, and the software will say, here are the genes that are being made in this organism, because here are where the red spots are. There's a lot of red here, so we made a lot of this gene. There's a little bit of red here, so we made a little bit of this gene. There's no red here, so we didn't make any of this gene. It's very, very easy to look at the entire organism, all the different proteins that are being made. So like I say, it's a high-tech way of doing a 2D gel electrophoresis. In this case, we're looking at messenger RNA. In the other case, we were looking at proteins. Now, we can also do the experiment that, like we talked about for the cancer versus a non-cancer cell. A person has liver cancer, you might open them up, you might remove the tumor, you might take some of those tumor cells and grind up and isolate messenger RNA. From that same person, you could take normal liver cells that are not part of a tumor and do the same thing. Grind them up. So you got one tube over here that you grind up and it's liver messenger, it's tumor messenger RNA. One tube over here that is non-tumor messenger RNA. You make this one red, you make this one green. Okay? You mix them all on the plate. Now you ask the questions, where do I see red? Where do I see green? Where do I see equal mixes of the two? Equal mixing of red and green gives you yellow. Okay? Trust me. Okay? You look at the microscope and you say, oh, wow, red. Red corresponds to tumor. If it's red, that means there's no green there. I know this is being made in a tumor cell, but not in a regular cell. I see green. That means there's no red in there. It means I have this being made in a regular cell, not in a tumor cell. I see yellow. It's being made in both. And so you can not only grade what's being made versus this versus that, but how much of one versus the other. Something that's more reddish or something that's more greenish tells me it's preferred in one versus the other. High-tech way, you can do this kind of an analysis in a few hours. It's really remarkable. Okay, that makes sense? Yes?
How similar do the sequences in the the grid, how similar do they have to be to the actual enzymes for them to stick?
Okay, so his question is how similar do the sequences have to be to those that correspond to the original genes? They are exactly complementary to them. Exactly complementary to them. Yes, sir, Matt?
As far as alternate splicing goes with RNAs,
yes?
Does that complicate things when they're trying to stick to the grid?
It's a good question. Generally, so his question is if you have alternate splicing that goes on, does that foul up the grid? If you know of alternate splicing going on, you probably would design a spot that had one splice versus the other splice versus the other splice. But in theory, yes, it could. And those are very interesting things when you see them. Some tumors actually do have different splicing than regular cells do, which is kind of cool. Okay, that's enough for biotechnology. We've gone very high-tech there. And I want to turn our attention to viruses. Viruses, okay? Now, viruses are health problems. Viruses, as I think you will see, are actually tools in some cases for putting DNA into people. It may be very useful ultimately in the future when we know more about how some of these tools work. So that it's important for us to understand then how viruses work and how they do what they do. Well, if we look at a virus, okay, this is a sort of a schematic of a virus. A virus has many different shapes, many different sizes, and in fact, many different types of organizations. So this only represents one type of virus. We're not going to worry about the different types. Okay? Viruses vary, for example, in the nucleic acid they contain. Some viruses contain double-stranded DNA. Some viruses contain single-stranded DNA. Some viruses contain double-stranded RNA. Some contain single-stranded RNA. Some viruses are in circles. Some viruses are linear. Some viruses are in pieces. The flu virus is an RNA virus. It's in pieces, for example. Okay? So there's a lot of different compositions of nucleic acids that can be among viruses. A given type of virus will always have the same general type of composition. So a flu virus will always have multiple RNA copies within its genome. Okay. An important component of a virus is what surrounds that nucleic acid. So there's always at least a simple protein coat around it, because that protein coat is designed to help protect the virus when it's out in the environment away from an organism that it's infecting. Okay? I told you how a human cell has 7 feet of DNA in it. And that tells you that human cells pack things pretty tight. Here is the head of a virus from which its DNA has just been released. Imagine how tightly packed that DNA was in the head of that virus. DNA can be wrapped up pretty tiny, pretty darn tiny. Okay? And let's see, I'll leave that alone. Okay. All right, here's a table that we're not going to pay much attention to, but I'll just point out. It's just illustrating to the different types of viruses, the different organizations that they have. You're not responsible for it. I'm just pointing it to you so that you see there's a lot of different stuff out there. DNA viruses, RNA viruses, etc., and causing a variety of problems out there. Viruses range in size of genome considerably. If we look at something like SV40, which is simian virus 40, it's a very commonly studied virus in the laboratory. This guy's pretty tiny. It's got about 4,000, 6,000 base pairs. Okay, we look at a herpes virus, which is much more complicated. It might have 250,000 base pairs. Big differences in size. Very, very big difference in the genomes that they have. The bigger the genome, the more genes a virus can have. The more genes a virus has, the less dependent it is on the host. But in every case, a virus has to use something of the host in order to survive. So viruses cannot live apart from the host. They have to use something from the host. And we'll talk a little bit about that. Okay. I like this figure depicting the life cycle of a virus. And it shows that a virus, in order to do its thing, has to first attach itself to a cell. And then it has to inject its nucleic acid in some way or the other. The nucleic acid, when it gets into the cell, then can do a variety of things. What was that? It may go to the nucleus. Some viruses replicate in the cytoplasm. Smallpox, for example, is a virus that replicates in the cytoplasm. Most viruses replicate in the nucleus. Okay? As it replicates, that means if it replicates, it either has to have its own replication system, or it's dependent upon the host's replication system. Whatever it does, its interest is in doing two things: making thousands and thousands of copies of its genome, and making thousands and thousands of the proteins necessary to encase that genome so it can go infect more cells. Ultimately, the virus assembles, it self-assembles using the proteins that it makes, and those proteins are little nanomachines. They literally come together to make this capsule that holds the DNA within inside of it. The infected cell ultimately bursts because it dies, and that causes the release of a whole bunch of new viruses that go out and infect more cells. Okay? This life cycle is a general life cycle. We'll see there are some variations on that for what a virus does. HIV is a very interesting virus, and I talk about it because it actually is useful for understanding what this class of viruses does and how this class works. Okay? HIV is an example of what we call a retrovirus. I've talked about retroviruses before. A retrovirus is a virus that has a genome of RNA. And a retrovirus goes through a DNA phase as part of its life cycle. So that means it has to go from RNA to DNA, which means it must have a reverse transcriptase. The reverse transcriptase of retroviruses comes in the genome of the retrovirus. So retroviruses have their own reverse transcriptase, the enzyme that converts RNA into DNA. Retroviruses have their own reverse transcriptase. That reverse transcriptase that we see in retroviruses, as I mentioned before, is not very good. It makes a lot of errors. It doesn't have the 3' to 5' proofreading. So when it's copying, it's making a lot of mistakes. And mistakes helps it to evade antibiotics that would otherwise kill it, antiviral drugs that would otherwise kill it. Okay. Now, the virus actually infects T cells. T cells are important cells of our immune system. And it infects them by attaching to a specific T cell protein called CD4. So the virus attaches to CD4, and the viral RNA is allowed to enter the cell as a result of this. Okay? Here's the viral RNA.